get going. We're ready. So we're back to the PowerPoint. So the neuromuscular junction is the place where the nerve, the axon, uh, forms a synaptic terminal. The ending of the nerve is called a synaptic terminal. It's where acetylcholine is released. Synaptic cleft is an actual gap between the nerve ending or the axon ending or the synaptic terminal. And that comes right up to the cleft. It releases acetylcholine, and the motor end plate has what are called acetylcholine receptors. There's an enzyme called acetylcholinesterase that is used to break down the acetylcholine, because once a muscle is stimulated to contract, the acetylcholine has to be removed so that the muscle can relax and be ready for the next contraction. So if you look at a colored image of a muscle axons and skeletal muscle fibers, you can see that the axon terminals are just above the surface of the sarcolemma, and any place the axon terminals form a synapse, and there's a motor end plate underneath each axon terminal. So the nerve is going to transfer an action potential, a nerve spike or nerve message, all the way from the axons and end at the axon terminal just above the synaptic cleft. It's also called the synaptic terminals. So the neuromuscular junction is the point where muscle fibers have contact with the chemicals released by the nerve. The synapse is an actual gap. So that's important because that's how the muscle translates the changes started by the nerve signal or the nerve spike or the action potential, how that message is converted into a biochemical message, and how that leads to contraction of the muscle fiber. So if you look at a sequential image of the synaptic cleft, the motor end plate, the sarcolemma of the muscle, so you can see that the action potential is moving away from the cell body. It's going to get to the synaptic terminals. There's going to be acetylcholinesterase, and there's also going to be receptors. So as the nerve potential or the voltage changes at the synaptic terminal, vesicles of acetylcholine are going to fuse and with the membrane, and acetylcholine is going to be released into the synaptic cleft. The acetylcholine binds to sodium receptors, and that opens the receptors or the sodium channels, and sodium rushes in from the cleft into the nerve. That sodium that enters into the inside of the muscle beyond the sarcolemma, that sodium causes an action potential or a positive nerve spike, or where sodium comes in, and that sodium spreads in either direction to cause the rest of the muscle to depolarize, and the acetylcholinesterase has to destroy that acetylcholine so that the muscle can relax and then contract as soon as possible. So the contraction process is cross bridges formed from the myosin to the active sites on actin. It's called the sliding filament process because the thin filaments slide past the thick filaments, and they are pulled toward the center of the sarcomere by cross-bridge attachment. So these bridges attach, pivot, detach, and return. The control interaction is what the calcium does to troponin and tropomyosin. So actin has active sites, myosin has cross bridges made up of the myosin tail and the myosin head. The sodium moving into the muscle through the sodium channels opened by acetylcholine, so they are called chemical gated channels. The sodium spreads out in either direction on all sides of the axon ending, right from the motor end plate, and that change in the charge inside the muscle due to sodium entering is going to make the myosin heads bind on the actin active sites. So what does that mean? So a resting sarcomere has the troponin and the tropomyosin covering the active sites on the actin. The myosin heads are energized by the breakdown of ATP into ADP and phosphorus. Remember, ATP is a universal carrier for the storage of energy and for muscles to contract. They have to have energy to allow these myosin heads to be energized. And when calcium is released, the active sites on the actin, the troponin causes the tropomyosin to rotate. And when those heads are exposed, the myosin sites or the myosin heads bind to the active site on the actin. So what's going to happen is that those myosin heads are going to move forward based upon the energy of the ATP. The ADP will be released, and after the myosin heads pivot toward the center of the sarcomere, that moves the actin toward the middle of the sarcomere, and the muscle contracts. Then as soon as that last step, as soon as if there's ATP available, the myosin heads are going to be recharged. So the whole process depends upon calcium being released to cause the active sites to be exposed. Once the active sites are exposed, the energized myosin head binds to the active sites. The energy allows the myosin heads to pivot toward the center of the sarcomere. As soon as the acetylcholine is destroyed and the myosin head has moved forward, it's re-energized and it's ready to bind to the active sites on the actin, and it's going to ratchet forward or move forward toward the middle of the sarcomere. And this process takes energy. That energy is supplied by ATP. When these myosin heads are reactivated, a chemical reaction separates ADP from phosphorus, and that energy is stored in the myosin heads. You can see that the myosin heads move forward toward the center of the sarcomere. That moves the actin overlapping more with the myosin, and the actin moves into the center of the sarcomere, causing each sarcomere to shorten. Okay, so here's the whole process summarized. Here's the axon, here's the motor end plate, which is the specialized part of the muscle sarcolemma. The synaptic terminal releases acetylcholine that causes sodium to move into the interior of the muscle. That sodium moves in both directions away from the synaptic terminal. And as the voltage changes, as the sodium becomes making the inside of the muscle more positive, that's called an action potential. That action potential moves down the T tubules. That causes the sarcoplasmic reticulum to release calcium. The calcium causes the active site exposure by binding to troponin and causing the tropomyosin to rotate and to expose the active sites on the actin. Once those active sites are exposed, the energized myosin head and tail bind and move with that energy toward the center of the sarcomere, and that causes the muscle to shorten. Contraction begins. Acetylcholine is removed by acetylcholinesterase. Calcium is recaptured in the sarcoplasmic reticulum. Active sites are covered. There's no place for the myosin heads to attach to the actin. Contraction ends and the muscle relaxes. So skeletal muscle fibers shorten as the thin filaments interact with the thick filaments. Calcium must be released to cause the active sites on the actin to be exposed to the myosin heads. Okay, so let's stop there. Let's go back here and stop recording. Stop recording.